I just want to say thank you guys for all coming out, those who are in person and virtual. And welcome to our fish population and bird or wildlife deformities or reproductive problems definition of experimental tests. Quite a mouthful. We shorten it to BUI 3 and 5. So is that no. It's some of these words.
Try again.
There we go. Okay. So we just want to start tonight off with a land acknowledgement. So the Essex region is the traditional territory of Caldwell First Nation, members of the Three Fires Confederacy, which is comprised of the Ojibwe, Odawa, and Caldwell peoples. We value significant historical and contemporary contributions of local and regional First Nations and all of the original peoples of the Wild North America who have been living and working on the land from time to time. Nope, this one, maybe.
Try again.
Okay. So, we're going to start tonight off with introduction. So my name is Sarah Liu, and I am the Remedial Action Planning Coordinator for the Detroit River Canadian Cleanup. I'm covering for inventory duties, so I'm not going to be here super long, but I will be here for the next month at least. And over there, working with computers is Laura Neufeld. She is the Community Outreach Coordinator and Remedial Action Plan Assistant for both IRCA and Detroit River Canadian Cleanup. Virtually, we have Shane DeSola. He is a legal toxicologist at Environment and Climate Change Canada, and he's going to be the one that did all of the work on BUI number 5, which is the Burger and Deformities one. We also have here Stephen Markowitz, and he is the Ontario coordinator for the Lake Erie Basin with the Ministry of Natural Resources and Forestries Canada. He's the scientist that did all the work on the BUI number 3, which is the fish population one. I also wanted to recognize and attendance we have Kevin Money, who is the Director of Conservation Services at IRCA. We have Basil White, who is the Program Officer for Environment and Climate Change Canada, and Ted Briggs, who is the Great Lakes Advisor for the Ontario Ministry of Environment, Conservation and Parks. And those 3 people make up the supervisory committee for DNRC, so they're overseeing the project. So just to outline the evening, we're going to start off with the introduction to Detroit River Canadian Cleanup. Then we're going to move into the bird or animal deformities or reproductive problems presentation. So that's the Shane presentation. We're going to do a quick Q&A period. So anybody who has any questions, they can ask them. Shane will be able to answer them directly. We're going to do a quick little 10-minute break, and then we're going to go into the fish populations beneficial use presentation. And then after that, we're going to do a final Q&A period, and that's it for the evening. Just as a reminder for those in person, there are refreshments at the back. There's water, soda, tea, all of those things. And feel free to get up whenever you want and grab a snack. And yeah, so just to start off here, an introduction to the Detroit River Canadian Cleanup. We wanted to take a little bit of history and a little bit of context. So the Detroit River, as you may know, is one of the busiest waterways in the world. And it is also notoriously one of the most polluted. And this is because of decades of industry and the creation of many shipping channels. And you've seen like videos on the internet in the past of like the river lighting on fire and things like that. And it's very evident that this is a pollution problem. So back in the 60s or 70s, people had started to notice that there were fish being washed up on the shores and these thick mats and algae appearing, and people started saying that the lake and the river were dying. And well, they weren't dying, they weren't held safe though. And actually that media outcry and that public outcry is actually what led to a legislative change called the Great Lakes Water Quality Agreement. And the Great Lakes Water Quality Agreement was signed by both Canadian and US federal governments in the 1970s. And what it does is it identifies 43 different areas of concern. So these areas of concern are areas where there is like hotspots of pollution sources, and they're areas that needed to be listed as an area of concern. Basically, they need to be repaired. And some of these are obviously we have the Detroit River, and as you can see here, there's a bunch of other ones as well. For example, the St. Clair River, Hamilton Harbor, St. Mary's. And there's all sorts of ones all along here. Another one actually that is quite close to home. In the US, they have the Rouge River. So that's like over around here. So moving on to what is the Detroit River Canadian Cleanup? So the Detroit River Canadian Cleanup was formed in the late 90s, and it's actually a partnership of these different groups all working together with the common goal of delisting this area of concern. Basically, we have a remedial action plan, which is the plan that outlines how we're going to delist this. And all of these different groups come together to scientifically work towards delisting. And we do this by looking at different beneficial use impairments, which is why you're all here tonight. So these beneficial use impairments, by definition, a beneficial use impairment is the use of a waterway that is negatively impacted by local environmental degradation. Things like being able to go to the beach or being able to eat the fish that you're fishing. You can see here there's 14 of them. These 14 BUIs, as we like to call them, are the same across all of the different areas of concern. So every single area of concern has different ones that are considered impaired or not impaired. And right now, we actually have only 4 that are still considered impaired, and we're looking at redesignating 2 of them in the coming months. So that's what this public consultation is for. So one big question is, when will the Detroit River be delisted? When is it no longer an area of concern under the Great Lakes Water Quality? Well, to look at that, we have to look at the 3 stages. So we have the stage 1, which identifies the problem, it scopes out what the problem is and the size of the problem. Stage 2, which is a report that we released in 2010, is actually going to go through and it scopes out the necessary remedial actions. So it identifies like what's still impaired, what's not, and how we're going to fix it. And then the stage 3 is going to be the final document. We obviously have not released it yet, but that's going to be providing evidence that all of the BUIs are not impaired, and from then on, the Detroit River can be considered delisted. Now, you may have a question that, well, the Detroit River is a binational river. It's shared between Canada and the US. So is the whole river delisted? Not necessarily, because the American side is also working domestically with the same 14 set of BUIs to delist their ag. So right now, we're only looking at the Canadian side, and the only 2 BUIs that we're looking at right now are the fish and wildlife and the fish populations. So the other thing that I want to talk about tonight, and the reason that you're all here, is the consultation for beneficial uses. So basically, we're recommending the following changes. We're looking at redesignating BUI number 5, which is our bird or animal deformities one, to not impaired, and we're looking at redesignating the fish portion of the benefit or the BUI number 3, which is the degraded fish and wildlife populations to not impaired. The way that this is going to work is we're going to give you presentations tonight, and tonight is the beginning of the public consultation period. This consultation period is set to last for 60 days. So if you don't get all of the questions answered tonight, don't worry. For the presentation, there's going to be QR codes up on the screen. There's one as well in the map page, that pamphlet, and they're posted on our website for those who are in attendance online, where you will be able to submit your comments at any point from now until December 4th. So that is pretty much all I have for you guys right now. If you want, so on your chairs, you will all notice the fact sheets. They were also sent out to those who attended online. They should be available on our website. If you want the technical reports, which is basically the long version of the fact sheet, we have some at the front for you to use during the meeting. You can scan this code to see them online. And yeah, without further ado, we will start the first presentation for BUI number 5. The too much talking.
Okay. Now we'll see if we can. All right, Shane, are you able to advance the slides? You're muted there, Shane.
Yes, as soon as you gave me control, my mute button just moved. But can you hear me now?
Yep.
Okay. Sorry, I am not really high speed, so let me know if you can't really hear me very well. Okay, so thank you much. I have some of my slides I can probably sort of skip past. Oh, don't seem to be able to advance the slides. It says I'm controlling. Okay, I don't know if I did that or not. So I don't have to go through this part very much because someone else just went through much of the description of the Great Lakes area concern for the Detroit River. For wildlife, the compounds that we're most interested in are things like PCBs and mercury because they biomagnify, and so you tend to get high concentrations in wildlife, particularly those which are high trophic level animals, but also pHs because even though they're not bioaccumulative, they tend to be fairly acutely toxic. Oops. All right, so we have two main criteria for the BUI 5 for wildlife reproduction and deformities. It's called wildlife reproduction deformities, but it's really just reproduction. We're not really assessing deformities. We have some deformity data, but it's not actually going to be part of the assessment. In any case, we have two criteria to determine if there's evidence of reproductive impairment in wildlife in the Detroit River. From historical contaminants. The first criterion is when incident rates of bird or animal reproductive problems in central wild species, which I'll show in the next slide, they do not exceed background levels at suitable reference sites elsewhere in the Great Lakes Basin or suitable inland control populations for a minimum of 3 years. We try to get reference sites as close as possible to the Detroit River without being in the AOC. Sometimes we're good at it, sometimes it's a bit further away. Second criterion, when scientifically defensible wildlife bioassays of integrated species confirm there are no reproductive problems and no significant toxicity from the water column or sediment contaminants or bioaccumulation. Since we're talking about wildlife, most of this would be through bioaccumulation or dietary sources of contaminants. And so, yeah, we're collecting our data to provide recommendations for current status of this BUI, that's impaired or not impaired. All right, so here are our main species for the assessment, as well as the years that we actually did the analysis, and lastly, the year we actually gave a final or draft report to the Government of Canada representatives of the RAP team, which then would send out to the rest of the RAP teams and any other stakeholders. So our 5 main species or group of species are northern leopard frogs, black-crowned night herons, snapping turtles, clean water birds, which includes both herring gulls, but that's a funny one, which you'll see in a bit, and double-crested cormorants, and finally tree swallows. So all of these animals are directly related to the aquatic environment. Some live in it all the time, some do not, but feed from it. So they have different aspects of the aquatic ecosystem, which I'll go to each one of the species to give a bit more sort of details. Hello. There we go. So for the first assessment, let's look at the northern leopard frogs. So northern leopard frogs, they're insectivores, but they do live directly in the water column. They do go on land, but they never really go very far away from water. And they eat primarily insects, many of which, of course, would be from the aquatic environment. So we did a series of exposures. We're looking first at embryonic exposure studies to assess both hatching success and deformities using the fertilized eggs, which we got from a reference site. And we exposed them just to water from Detroit River AOC sites. And then 2010, both water and sediment from several sites on the AOC. So you can see the locations of the sites in both these sort of maps. And also we looked at legacy contaminants in young of year. We used whole bodies from collections from 2008 and 2009, and we compared these concentrations to those on reference sites in Lake Huron. So you can see where we got the frogs on the map on the left. We actually wanted more sites, but sometimes it's kind of hard to find frogs on land. It was easier to get the sediment water to do exposures. Let's see. All right, so here is the data we got from these exposure studies. There's not a whole lot to say here, as you can see why. The mean hatching success was 97% or higher in the water exposure study and 85% or higher in water sediment study, regardless of where we actually got the sediment and water from. So there were clearly no differences in hatching success among AOC and reference sites. I mean, these are really high hatching success. Even 85 is still pretty good. So, I mean, we didn't bring these embryos all the way to, to adult frogs, but we went quite a ways along the development. So it indicates a very good ability of the eggs to hatch and also become free swimming tadpoles at the AOC sites, regardless of the degree of contamination. Now, we also got a whole body of frogs to measure contaminants. First of all, some PCB concentrations of frogs are not actually consistently higher in AOC frogs versus reference frogs. I mean, some sites were a little bit higher, but only certain years and not other years. But there certainly was no consistent in comparison with the reference site frogs. But yes, the highest some PCB frogs were from Turkey Creek in 2009. Now, there was some remediation going on upstream, which could have possibly caused a plume of PCB-laden sediment to be released, which maybe one reason why is 2009, the levels were higher than were in 2008. Because of this, we went back later on, got turtle eggs from along this site in 2014 and 2016, which I'll talk about later. Most importantly, though, some PCB concentrations of frogs were below those considered to be toxic. I mean, these are, generally speaking, pretty low concentrations. These are not high concentrations nasally matter. So that's pretty good news. Based upon these endpoints, we recommended that there's no impairment of reproduction associated with contaminant-induced effects in the Trick River AOC. Now, not to say there aren't other problems, but most of the problems are probably more along the line of habitat, but do not appear to be any problems due to contaminants. We also assessed black-throated night herons. Black-throated night herons, these birds, they feed both terrestrially but also aquatically. They will eat voles and mice and that sort of thing, but they tend to go along the water's edge and they eat fish and frogs and other aquatic animals, large insects, that sort of thing. There are a number of species of clean water birds that used to nest on the Canadian side of the Trick River. We discovered this colony in 2009. It's not a huge colony, but there was a fair number there. It was this was on Turkey Island, which is southeast of Fighting Island, and so we started assessing in 2009, and we also did in 2011, and we used a reference colony called Nagawasaga, which is in Georgian Bay. It's a reference colony, and we assessed contaminant burdens in the eggs and nesting livers at the sites, and also look at fledging success and hatching success. All right, so the number of eggs hatched was lower at the AOC colony, Turkey Island, versus the reference colony, which is Nagawasaga Island, 2009. That was similar between colonies in 2011. However, most of the reduced hatch success was due to egg loss during incubation. And of course, egg loss is not from contaminants. We believe there were actually predators. It's not that unusual for predators to, raccoons, foxes, or whatnot, to take eggs from their heron nest. So we think that a reduction in the number of eggs hatched in 2009 was from predation. If it was contaminants, the eggs would just simply not hatch, and they would still be there sort of adult, but we didn't really see that, suggesting that contaminants was not the issue. Now, the numbers of nestlings fledged per nest, which we call productivity, was lower at the AOC colony versus the reference colony in 2009 and 2011. However, productivity was high at both colonies in 2011 versus 2009, whether we're talking about the reference colony or the AOC colony. Now, the big reason why the number of nestlings fledged per nest in 2009 was low, which is simply because there were fewer eggs, because the eggs were probably taken by predators. In any case, you can see on that graph a lower threshold for a stable black-throated night heron population. So this is the productivity which we have to maintain in order to have a stable population over quite a number of different years. And you can see that all except for 2009, they were actually higher. As it was lower in that 1 year because of predation. Now, I'll talk about something else in a second. So we also assessed contaminants. So PCB levels were definitely higher in the eggs from those in the AOC colony for a couple of years. Having said that, they were quite a bit below those associated with decreased hatching success in birds. So the highest concentration on average is about 1.7 parts per million. To give you an idea, we have data in herring gull eggs throughout all the Great Lakes, and you'd be hard pressed to find any gull egg with PCB levels that low. So yeah, the PCB levels here are not remarkable at all. Mercury levels were, however, were similar between the two colonies in both years, and they as well were below thresholds associated with adverse effects on reproduction. Now, 2 eggs of 22 at the Turkey Island, which is the AOC colony, in 2 years exceeded the 0.6 microgram per gram threshold to be protective against adverse reproductive effects in birds. Now, this doesn't mean there will be, toxicity. That's just when you start, there's a slightly higher risk that may happen. So even though it's above this threshold, it didn't go very far above it. And so we think it's unlikely causing adverse effects. So those are means that those are just sort of highest concentrations. So as I said, most of these stressors seem to be predation or disturbance, which reduced hatching success in 2009. Now, we've had problems with colonial water birds. I'll just mention here, I'll probably talk about later on. The birds disappeared. We were going to do another year of assessment, but the colony disappeared. But this wasn't the only colony that disappeared. Pretty much, we don't really know why, but every colonial water bird in the Canadian side disappeared in the same year, whether we're talking about ring-billed gulls, herring gulls, or black-crowned night herons. And we even saw some disappearance of Canada geese. But that does not appear to be due to contaminants. In any case, our recommendation is that there's no impairment of reproduction associated with contaminant-induced effects of black-crowned night herons in Trick River AOC. Next, snapping turtles. These are almost entirely aquatic. You will sometimes see the females drag themselves on land to lay their eggs, and if you're lucky, you can see little hatching turtles after they hatch, wandering around trying to get to the water. But other than that, they spend 99% of the time in water. They're highly aquatic, and they are piscivorous, but they're very much generalist feeders. They'll eat on algae and plants and invertebrates, fish. They'll eat young birds that they catch, occasionally snakes, frogs, that sort of thing. So they'll eat pretty much anything they can get their little, mouths on, or as adults, it's very big mouths on. We're able to get these a bit further inland along some of these rivers or creeks, such as Turkey Creek and Cannon River, and we also found some on the Little River. We did 1 year get one from Ojibwe Park as sort of an inland reference site. We weren't particularly targeting it, but someone said, "Hey, we got clutch here. You want it?" So we did grab some eggs, but I'll get it in a second. So what we did is that we collected eggs from all these sites to incubate them to assess hatching success and deformities. So what we tended to do is collect entire clutches of eggs from all these sites from 2014 to 2016, and the clutch size probably ranged from about like mid-20s to the high 50s or low 60s. The average clutch size is probably about 38 to 40 eggs per clutch. And we took 5 eggs per clutch for contaminants, 2014-15, and we got hatchlings analyzed in 2016. But most of the eggs that we did not actually use for contaminants. We raised up for hatching success and deformities, and those ones we actually afterwards released them back at the site that we actually captured them. And then for comparisons for reference sites, we used Long Point on Lake Erie. So the hatching success from the Trick River AOC was 90.1% or higher in regards to year, and was very similar to that of Long Point in all 3 study years. So this is pretty good hatching success. Typically, snapping turtles occasionally have, because the clutch is so large, they occasionally have these eggs which are not actually fertilized, and there's no development whatsoever, at very low levels. So this hatch success is not unusual. As I said, it's actually pretty good. Now, we also measured contaminants in each clutch, as I mentioned. So we did see that there are PCB levels in Trick River AOC eggs versus reference sites in 2014, 2015. There's no differences in mercury, however. Now, the one site we got from Ojibwe in 2014, the concentrations were, really low. both for mercury and for PCBs are actually lower than long point. But I said that's just a single clutch, so it's kind of hard to say very much about that. PCB concentrations were well below those associated with decreased hatch success in birds. There's not as much information in turtles, but there's some correlative information suggesting that for turtles, again, they should be unaffected by PCB levels at these low levels. Mercury concentrations were also below those associated with toxicity. That's decreased hatch success in turtle eggs. It's about 3 micrograms per gram, or 3,000 nanograms per gram, and we only had like 200, so it was well below that sort of threshold. Now, remember how I mentioned that 1 year some frogs had somewhat high levels at Turkey Creek. We think that was a radiation upstream. So we had some data in turtle eggs from the same general area back 2001, 2002, and 2014, 2015. This is a bit after we collected frogs. And we analyzed, for PCBs, and it appeared to be a reduction of about 50% of PCBs in the Grand Marais drain, downstream of the Grand Marais drain remediation sites, between 2001 and 2002 versus 2014 and 2015. So these turtle eggs were not collected at the Grand Marais drain. It's actually further downstream, but they will be impacted by any sort of sediment being released. In any case, this suggests a decrease in exposures turtles that's consistent with removal of PCB contaminated sediment. Sometimes what happens when you disturb sediment to try to remediate it, you get a short-term plume of PCBs being released, but very soon after the levels sort of drop as it goes back to normal. But the worst of the PCBs, of course, are removed. So our recommendation is that there's no impairment for reproduction associated with the contaminant-induced effects in snapping turtles in Turtle River AOC. There are challenges, of course, for turtles in this area as well. There are other areas such as cars hitting the females nesting, and there can be a lack of nesting habitat for females. But we saw no evidence that PCBs or any other contaminants were actually causing any impairment of reproduction of snapping turtles in Turtle River. Okay, other clean water birds assessment. So yeah, as I mentioned before, we had some difficulty in terms of getting clean water birds on the Turtle River because there were some there and they all sort of disappeared. The black-horned herons took off, and same with the green-billed gulls, which we don't normally use for assessments in any case because they eat so much terrestrial diet, so they're not that good at assessing aquatic ecosystems. But herring gulls are, but they all disappeared. And this kind of left just a couple options. First, there was a colony, we call it that, of double-crested cormorants. We call it Shag Island. And just to be clear, shag refers to a British term for cormorant. It's this little tiny island northwest of Fighting Islands, actually the US side, but just barely. You can see on the image in the bottom right corner, it's basically just a couple dead trees and an old tower. So there was only like 30 nests or so right there. So we collected eggs from this colony in 2016, 2019. And we also conducted contaminants of cormorant eggs and embryos. There were some herring gulls downstream of the AOC, so it's maybe a little bit controversial. So there were no herring gulls nested in the AOC because I mentioned all disappeared from Fighting Island, but there were a few islands which are immediately downstream of the Detroit River and certainly well from the plume of sediment and also contaminants from Detroit River. The contaminants you see, Middle Island, Chicken Island, the Middle Island, are all consistent with what you expect to see from the Detroit River. So we're using it as a surrogate, even though technically they were not within the AOC itself. So we're looking at productivity at these sites and contaminants. All right, so embryonic viability in double-crested cormorants. That's what DCCO stands for, double-crested cormorants. We looked at embryonic viability in these eggs that we collected. And well, you can see that figure where Shag Island, Middle Sister Island, which is downstream, of course, and Black Rock and Walk Island, which was the reference site. And I mean, they're basically identical. They're 83% pretty much every site, which is there's no difference among sites, but also is also really high. So we had no evidence of reduced viability of cormorants foraging within the AOC. So for herring gulls, looking at productivity, what we do is we put these enclosures around some of the nests so the chicks couldn't just wander around, and we assess them over a number of periods of time. And then at the end, we looked at how many chicks there were within each enclosure. So yeah, the time that we used was when the chicks were 21 days old. At that point, they're not quite ready to fledge. They couldn't fly yet, but they get kind of close-ish. So we got estimates at AOCs and reference colonies, and you can see that in the graph on the bottom, that little red bar, that is a lower threshold for stable herring gull populations. You can see every colony we did for every year was well above this. So there's no expectation that productivity is too low to maintain healthy herring gull populations. Looking at burdens of PCBs, both embryos and eggs of double-crested cormorants on Shag Island, Middle Sister Island, and the Black Rock Mohawk Island, which is the reference site, the one in green. You see Shag Island is below it. Middle Sister Island, which is outside the AOC, but within sort of the influence, if you like, of the Detroit River AOC, it was barely, but nonetheless below the threshold of 8 micrograms per gram, a threshold for decreased high success for terns and cormorants. So that was for the embryos, and if you look at the eggs, they were fairly well below that for Shag Island, Middle Island, and Black Rock. Looking at mercury, it was unremarkable. Mercury is not, for wildlife, is not a big deal for the Detroit River, no matter what. So looking at, with one exception, which we'll talk a bit, you can see that all the colonies were below each of the threshold for mercury levels associated with adverse effects on reproduction. Now, we did see some higher deformities at Shag Island cormorants, but as I said, that the deformities were not actually part of the assessment for health of clean water birds in the or any wildlife in the Detroit River AOC. So our recommendation is that there's no impairment for reproduction associated with contaminant-induced effects in clean water birds nesting within the Detroit River AOC. As I said, clean water birds disappeared, but that would not be a contaminant issue, and we're not really sure why they were these colonies, which have been nesting there for decades, have all disappeared simultaneously. Tree swallows. Now, most people, well, many people would really think of these as aquatic. Of course, they don't swim, they don't go in the water, but tree swallows eat primarily emergent insects, and the colonies tend to be best along wetlands because that's where you get most emergent insects. So they often eat mayflies or other sort of water-living insects that will emerge from the water and then fly off. Then the tree swallows go and snap them up. And they're pretty handy because we can, unlike the other species that we've mentioned, we can dictate where they nest simply by putting up nest boxes, which they appreciate. So we purposely put in nest boxes at Sandpoint Beach Park, in Shanfeld Shores Park upstream, and then near Winter Salt Mine, River Dance Park, and finally White Sands Conservation, which is downstream of Fighting Island. And then so this is very convenient because we put the boxes exactly where you want, and basically if you build it, they will come. So we were assessing reproductive success in tree swallows, and we use eggs for contaminant analysis. For all of these sites, we put up about 25 nets boxes. We monitor each year for 2016 to 2019. We were going to do it 2020, but because of COVID, that sort of made things difficult, so we didn't do it in 2020. Anyway, so the upstream sites are the reference sites are the Sandpoint Beach Park and Shanfeld Shores Park, which we kind of treated as sort of like one site. So here's the data on hatching success and fledgling success. Hatching success range from 80% to 100%, and fledgling success was greater than 67%, with one exception in 2016 at the, of course, the reference site, Shanfeld or Sandpoint. That was pretty certain was due to predation, and since then we put these nest guards on the boxes, which dramatically reduced predation. There's a number of things which, of course, eat tree swallows, such as snakes and small mammals, that sort of thing. But yeah, after the predators had put up, the predation stopped being an issue. We did not, other than that 1 year, one site at the reference sites, we did not see any significant differences in reproduction among sites, either for hatching success or fledgling success in the 4 study years. Looking at PCB levels in the eggs, we didn't really see any differences in some PCBs in the eggs among years or among the 3 sites, and the concentrations were well below those associated with reduction in hatching success and tree swallows. On the US side, they have been putting tree swallow boxes everywhere, so we were able to compare our results to the US Fish and Wildlife projects that have been going on in the Great Lakes area is concerned. But so yeah, these concentrations are unremarkably, they're fairly low. Mercury. We did see some differences in eggs in 2 or 3 years, but with that one exception, which we'll talk about later. The differences are very small, and some years there weren't any differences. The highest concentrations tend to be the river dance or winter salt. You do see, of course, in 2019, this really big blip. So although the majority of eggs were below the threshold associated with toxicity of mercury, 1 egg sample from Riverdance of 22 samples at site had very high levels of mercury of 1.3 micrograms per gram, which is slightly over twice the threshold. However, we didn't take all the eggs from that clutch. We left the rest there, and all the other eggs that we didn't take the contaminants, they all hatched. So it all seemed to be fine, despite that. So we don't know why that one nest had high levels. It's not really indicative, I don't think, of that site or the other clutches that year. It's basically mercury was pretty much the same that year in the birds, except for 1 clutch. So why it's high, no idea. Maybe a thermometer or something like that. I don't know. So our recommendation is that there's no impairment for reproduction associated with the contaminant-induced effects in the tree swallows nesting in the Detroit River AOC. All right. So here's sort of like putting it all together for the BUI 5, the wild reproduction deformities. But as I said, it's really just reproduction, not deformities. The first criterion with incidence rates of bird and animal reproductive problems in sentinel wildlife species, these 5 as mentioned, do not exceed background levels at suitable reference sites. So we're looking at average incubation of eggs, success intrinsic factors, and early embryonic development is usually the most sensitive life stage. We did this for cormorants and turtle eggs. So we've got 2 very different, sort of species. Look at hatch success, and there was no difference between AOC reference sites. Frog eggs are again very, very different, and these was following both exposure to water and water sediment among the different sites. So basically, we're looking at contaminants which were in both cormorants and turtle eggs, which were transferred from the female, maternal transfer is called. The frog eggs in direct exposure through water and sediment. And we did field studies of wild populations, both of white, black-eyed night herons and tree swallows to assess productivity. And we didn't really have any issues except for that 2009 when hatching was low, but we think that was due to predation. And the productivity estimates for herring gulls were definitely high enough to contain the stable population. So the only issue here is that the clean water birds all disappeared, but again, that is not a contaminant issue, that's a non-contaminant issue. For criterion 2, using scientific defensible bio, we tend to focus on assays which are associated with effects that can affect population levels. So we didn't do gene expression or anything like that, which is really hard to assert whether it has any real problem with population levels. But looking at hatching success and productivity, which clearly do have population level sort of effects if there's any impairment. But the concentrations that we found in pretty much everything except for that, 1 weird high level of mercury in 1 clutch in tree cells in 1 site 1 year, we're basically all below those associated with adverse impacts of reproduction from the vast majority of samples. So just that tree cell egg. Oh, and also 2 of the black-eyed night heron eggs. But again, the rest of the clutch seem to be fine. And in a case, had success was or was not reduced in that year for those sort of high levels of mercury. So when assessed against the listing criteria for Detroit River AOC, we seem to have met it for the clean water birds, for the black night herons and cormorants. We met it for the snapping turtles, tree swallows, northern leopard frogs, and so, oh, also, I should also say, under clean water birds, black night herons, and cormorants, and herring gulls, that should have been there as well. So there's little evidence for impairment of reproduction associated with contaminants with AOC, so a recommendation for the status to be changed to not impaired due to contaminants. And I think I'm done. Hopefully you can hear me well.
Awesome. So if anybody has any questions, we're going to do a Q&A period now for this BUI. So yeah, if anybody has any questions online, just feel free to unmute yourself and ask. All right, I can see. Oh, sorry, Shane, would you be able to stop the control for the screen?
It may take about half an hour to figure it out here. Shoot, where's, sorry, options. Did that work?
No. It's next to the annotate button. It says remote control. I think you just have to like hit stop.
Sorry, where is it? Sorry, it's beside what?
Beside annotate on the top bar for Zoom.
It says I already pressed to give up remote control. Right now it says.
There we go. Got it now. Sweet. Okay, we are back. All right. So I can see we have our first question here online from Robert Burns. So if you want, feel free to say your question.
Yeah, hi, Robert Burns. I'm with the Detroit River AOC on the American side. Shane, I had a question in regards to deformities, and you mentioned that there was some deformities noted in some of the cormorants. I believe it was on Middle Sister, which was one of your sites, but you said that's not an impact. I kind of missed the connection there because my understanding is we're looking at reproduction and deformities. Can you explain that?
Probably other folks who can probably explain it better. For the BUI, it was determined that they're all going to focus on reproduction, and they were not using deformities as part of the assessment. And I believe the reason is that there was very little evidence in the past that deformities were a problem. It was just reproduction, so they're therefore not using that as assessment for whether or not there's any impairment of wildlife at the AOC. So that's not decision on my end. I just give data to the Great Lakes folks. There was hopefully someone else here can explain better. April Wright is probably good at explaining, but I don't think she can actually talk, unfortunately. So it was not really used for the assessment. Having said that, although we did see an increase in deformities in cormorants from one colony once, it was not particularly high. And we didn't see very much of a deformities for any other sort of species that were monitored.
Was that cross bill, or I was curious on what the deformity was?
Oh, I don't think it was. We have data somewhere, but unfortunately I don't have it in front of me. I believe it was either edema or there was some problem with development of his stomach.
Okay. Yeah. Again, I was just curious. Like I say, your determination is a little bit different on your side than our side, but the state and EPA are the ones that define what our criteria are for our BUI removals, and the fact that deformities is part of ours and is kind of a driver on our side. I was just curious on how it got excluded on your side, but if somebody somewhere made a decision that you were going to exclude deformities and concentrate on the reproduction part of it.
Right. And the interesting thing is, though, it wasn't Little Sister Island that the eggs were from. I believe that was from the. So I wish I had all the data in front of me. I'm at my mom's house right now. I was coming back from a conference. I couldn't make it back to my work building before this meeting was going to start. I believe, however, the deformities were technically on the US side. That was from Shag Island on the northwest side of Fighting Island. So technically those higher deformities were on the US side. I don't
Okay.
know if that makes any difference to you or not, or whether that information is useful to the US side or not.
So you said the deformities then were on Shag Island, not on Middle Sister. I maybe I mistook that.
I believe so, yes.
Okay, I know exactly what you're talking about. I know the island very well. Okay, thank
Okay.
you. I appreciate it.
Well, you know what, I really should go and check it up. I can't do it today, but I will go back and make sure I got the right site where the deformities were slightly higher. And I don't know how to get the information to you, but I guess I can send it to the host here. And
Yeah, they have our contacts for our site. Yeah, I'd just be curious on, again, it's more of a formality on making decisions on ours, because we're, our site's pretty specific on what our criterion are, and we don't make that local. We don't make changes locally. It's something that we work with. But yeah, I'd just be curious on where the decision came from to just concentrate on one aspect as opposed to the other.
okay,
Thank you. I appreciate it.
I'll send what I can to the host. And also we have all these reports, which I believe are available to anyone. So we have all reports for all these species. So we can probably get a PDF of them.
Thank you.
Yeah.
Okay, awesome. So yeah, Robert, we will make sure to follow up with you. I believe I have your email from the Detroit River PAC meeting, so I will shoot you an email once I get that information from Shane.
Thank you.
Yep, and then does anybody have any questions online or in person? Any other questions? I am not seeing any. So we will jump into taking a quick break, and on the screen there you can see there's QR codes. So if anybody has any questions that they forget or they think of right away, feel free to scan that. You can submit comments online from there. So yeah, I don't know. We will reconvene in 10 minutes. Enjoy the snacks and talk to everybody, and yeah. There we go. All right, we are going to get started again. So next up is our presentation for BUI number 3, and Stephen is going to be the one presenting for that. So I will give you that remote. It should.
Where that was on.
There. Okay.
Or was that you? That was me. Try it again. There.
Okay. There we go. We got it.
You want me to stand up to here?
I mean, you can do whatever you want. The cameras can see you whether you're standing or sitting, so it doesn't really matter.
Generally, I move around a lot, like standing up to the presentations. I know some people came in after the introduction, just to remind everyone. My name is Stephen Martaglia from the Canada Ontario Agreement Coordinator, formerly Erie Basin for the Ministry of Natural Resources and Forestry. As part of that role, I actually sit on all 3 areas of concern. So Detroit, St. Clair, and Niagara. And I have to admit, for the fish populations work that we've done here, this is the most data rich of all of the 3 areas of concern. It has been sort of the easiest to do an assessment on because of that. So it was really nice. It was probably in large part due to the university being here and lots of great work that's been done over the years from that group, Merca, some of the other people in the area. So unlike Shane's previous presentation, I actually didn't do any of this work myself. Most of this comes from a literature review we did existing, sort of a lot of existing studies to pull that all together to tell the story with respect to the fish populations on the river. Sarah did a fantastic job when we first got here, describing the area of concern. Not going to go into a whole lot of detail there to repeat that. But just a couple of things very specific to fish community. So that industrialization and urbanization that occurred in this area during the early part of the 19th century, that really had some fairly significant effects to the population. There was a point and non-point pollution, the destruction of wetlands, hardening of shorelines, which I'm sure if anyone's been out on the river, you can see. And probably the most significant in this area, the dredging and channelization of those shipping channels I'm going to show you a picture in just a minute to show you the scale of which this was actually done. So Troy, Sarah covered it already. It's one of the original period concerns in 1987, with fish populations being one of the 4 remaining BUIs. So here's that image of the amount or the significance of that habitat destruction that occurred with some of the shipping channel creation. So on the left here, it's your left. This is the creation of the Livingston Channel, in which they actually coffered the dam out, and I don't know how they did that. I assume dynamite blew out large sections of the bottom of the river to create that depth for the large vessels needed. They literally cut islands or created in half or created new islands with the spoiler the material that came out of here. So we have a very altered landscape here. This did lead to some real concerns. So to give a bit of a timeline for fish populations and the fish populations beneficial use and impairment here. So a lot of that destruction happened and that degradation happened in the early 1900s. We listed the area concerned in 1987. That stage 1 report came out in 1991. At that time, the fish populations were actually not considered as impaired. And it was noted that the river was supporting a vibrant and diverse fish community at that time, and it wasn't felt that they were actually impaired. Moving forward or jumping forward a little bit to 1996, there was a bit of a draft report here that was still sort of indicating that fish populations shouldn't be impaired, although that wasn't mutually agreed upon at that time. And in fact, this report didn't actually ever get finalized, I think in part because of that. The next time you see some reference to fish populations was within the 1991 evaluation. And this was the first time we actually saw fish populations being listed as impaired. The reason for this impairment was cited for contamination of PCBs, pH, and heavy metals within fish tissue. And that was sort of a large part of why that shifted at that time. Moving forward into 2006, it remained impaired. The rationale for why it was remaining as impaired had shifted again. So again, not as heavy of a focus on those heavy metals and contaminants, sorry. And more of a focus on a shift in the fish community, thinking that there was more invasive and benefit fish species at that time. And that designation is more or less remaining through 2010, right up to the present time. One of the challenges we've had with the fish populations BUI is our delisting criteria, and we'll touch base on it, but it was a very all-encompassing statement. We wanted healthy and self-sustaining fish populations. What that means to different people, what that meant to different people, including technical experts, was different between those individuals. So we were really lacking solid criteria for being able to evaluate what a healthy fish population and a healthy and self-sustaining fish population is in the context of the area of concern, and in a way that we could actually measure it. So in 2021, 2022, a group of technical experts came together, looked at the data we had available to us, which represented what we could actually collect within the system, which is a really important thing with all of our connecting channels. And we redevised our criteria to be SMART, or specific, measurable, achievable, relevant, and time bound. So sort of narrowing in what we can measure to evaluate our delisting criteria. So overall, our delisting criteria through all of this time frame hasn't actually changed. The goal has always been that the environment should be supporting self-sustaining and healthy communities of fish. We've changed that to indicator fish species slightly because we needed to sort of narrow it down. Those being identified as originally as walleye, bass, lake sturgeon, brown, bullhead. And then we created these these are the 5 sub criteria. So 1 being fish communities in coastal wetland areas are comparable to fish communities in unimpaired coastal wetland. 2, the number of adult lake sturgeon in the system are greater than 7, at or greater than 750. I'll touch base on where some of this stuff comes from as we walk through it. So don't worry, I'm gonna get there. And that, 3, that there's evidence, no evidence of identification in the fish community. 4, creel and catch per unit effort survey, so this is angler survey of walleye and bass, indicating retention are at or above a long-term average. And the 5th one, 5th and final one, is that there is evidence of ongoing or increased spawning since 2006. So we're going to just step through each, and I'm going to present to you the information that we found that supports each of these sub-criteria. So sub-criteria number 1 is that wetland and fish communities are comparable in the area of concern and to a sort of a reference condition. So that reference site is actually multiple island vegetation marshes. Located at the south end of St. Clair River or the north end of Lake St. Clair. For this, in our review of the available literature, we found one purpose-built, I'm going to call it purpose-designed study to evaluate this. This is out of John Whitwood's lab from the Department of Fisheries and Oceans, in which they used an index of biological integrity approach to evaluate fish communities in the Victoria River, fish communities in the Walpole Island Delta. So IBIs, or index of importance, takes a bunch of different metrics around the fish community and kind of matches them into one level, and that it allows you to sort of do like for like comparisons because you do your sampling the same way. Overall, this study caught over 4,000 fish from 49 species within the river. Those scores, those index of relative importance scores, which is kind of like an A, B, C, D, or E in school, were the same, were more or less the same in the Victoria River as they were from Walpole Island, from the Walpole Island Delta. John and his colleagues were concerned that because we mapped all of these metrics together with the IBI approach, that might be masking some important differences in fish community assemblages. So through our research, we actually found multiple other studies that span well over a decade. I think the earliest one is 2010, right up to 2021, that were different studies looking at fish communities within the Victoria River. And within each of these studies, there was absolutely no evidence of any type of impairment. So we feel that the Midwood study combined with these other studies really do not show any signs of fish community impairment with the Victoria River. We feel that this sub-criterion has been met. I don't ever want you to just believe me. I'm going to try to show you some data too. So these are the index of biological integrity scores for the Victoria River as well as the Walpole Island Delta. So as you can see, the average IBI scores for both are almost identical, not quite, but almost identical. But what's also really important here is those whisker bars that are there talk about the variance. So not only are the averages similar, but the variability between the sites that they sampled are also similar, which actually speaks to the diversity within the Victoria and the island are similar as well, which is really important for different species. So that's sort of more evidence that things are looking good for wetland fish communities. Moving on to sub-criteria number 2. This is the number of adult age sturgeon within the river are at least 750. I told you I'd tell you where the 750 came from. That 750 actually comes from a lake sturgeon restoration plan out of the States. 750 was the number through genetic analysis, and I think professional opinion factored in there a little bit as well, below which they would be concerned of long-term population structure issues, so loss of adaptation of alleles, so their ability to adapt favorable traits might be impaired. So genetic depression first, but a lot of them. There's been, thanks to our U.S. Fish and Wildlife Services folks on the U.S. side, there's been an ongoing long-term study for quite a while now that have been tagging individuals and then releasing them and then recapturing them. So these are called non-recapture studies. So their first, so both of these papers or reports come from that ongoing study. The first report, with early data coming in, was estimating the population at 4,422 individuals. There's a bit of a caveat to these numbers. They include juvenile, they include everything, so adults and juvenile. Most of them are adults, though, just for record. And then the second study is a couple years later, so this one's actually, I think it might be out now. I have to check with Justin on this, but it's in review or almost out. An updated estimate using the same study design, same individuals, and the population estimate is actually grown to 6,406 to be. So well above the 750. Even if half of those were juveniles, which it's not, still well above the 750 threshold that was established in that restoration document. But the great news with this is that year over year, catch rates continue to increase, which is actually going to, or what's actually suggests that this population estimate is going to continue to grow as more and more data comes in. We're not going down, we're moving on. And a large part of this is attributed to a large part of the rebound surge within the rivers attributed to some of the reef restoration sites, which I think Kevin's been involved in some of them. Actually, I'm pretty sure Kevin's been involved in some of them. So, you know, there's one around Fighting Islands, and a lot of them are on the US side. Fighting Islands on our side, right, Kevin? Fighting Islands on the side. So good news on the lake sturgeon front. Again, we feel that the sub-criterion has been met. Moving on to sub-criterion number 3, this is the one that there is no evidence, so it's a negative here, bentification of the fish community in the Victoria River. So bentification is most commonly looked at when you're thinking about food webs. And in the Great Lakes, it's commonly associated with the invasion of racemid mussels, which is the aquatic mussel, and then the subsequent invasion of branchiolites. So that's really what we're talking about here. With respect to the fish community and in the Victoria River, so we're concerned about this shunt and driving of the fish community from pelagic fish, things like there's not many salmon, things like salmon, trout, whitefish, things swimming up in the waterfall, to things that are going to swim near the bottom. So things like branchiolites and suckers and carp. So again, large part big thanks to Environmental Climate Change Canada for funding research again from another GMO scientist. That's Lauren Curry's work. They actually went out and specifically looked at this. They specifically went to look for evidence of bentification in the Victoria River. So those bottom dwelling fish species, the pollution tolerant fish species, ones that would be indicative of degradation. What they actually found was that there was more pelagic fish species than benthic fish species within the Victoria River. With benthic fish species, we get about 33% of the fish community. And this was actually lower than in many of the other areas of concern, some of which fish populations are not considered impaired. So boat electrofishing catch-created effort, that's what CUE stands for tolerant invasive species, which again is one of the key metrics here. It was lower than it was in some of those healthy, those areas of concern which fish populations are considered. Again, we feel we've met this status, but again, you don't have to believe me. I'm going to show you some of the numbers here. Not the prettiest thing in the world. But here we can look at our CUE or catch-created effort. This is boat electrofishing. One time it's been way here. So we see the Victoria River catch-created effort compared to the Niagara River, the St. Clair River. Both of these rivers, the fish populations are at a very similar state here, where we have the information that we believe they're unimpaired and progress is being made that way. Bay of Quinte, it is not considered impaired, which is in Lake Ontario. Hamilton Harbor, fish populations here are considered impaired, with identification. So we would have expected, if we saw evidence of identification, we would have expected a number much closer to the Hamilton Harbor number with our history unit effort than we're seeing currently. And again, they used the time period of 2010 as sort of the before and after snapshot, and there wasn't really much shift in those pollution tolerant, benthic invertebrate species compared to other benthic species, including species like walleye, yellow perch, just to put it in context, versus non-benthic fish species. So the composition is very similar between the two time periods. So again, we feel we've also met this subcriterion. So this is where my ministry gets to come in. I can highlight some of my ministry's work here. So this is our creel and catch treated active survey, so angler surveys for walleye and bass. Within the river, the at or above those long-term averages. Just a couple notes here. This took a lot of work to get to. Why did we land on what we're talking about smallmouth bass here? So this is one of those things that we had to have long-term historical data on as well. So that's part of the reason we landed these two particular indicator species because we have long-term data sets. In fact, my creel through our creels from our ministry are back to the 50s and 70s in this case. So that's really why we focused just on these two species because of that historical data that we had. So so creels from the more recent time periods, so the 2002, 2009, and 2015, were in fact above the long-term averages for both species. Again, creels are finicky beasts as far as an assessment tool goes. So we try to confirm it when we can. Luckily for us, we can turn to our counterparts and see what they were seeing in their creels, which should have a very similar results as well. The other survey we had within our ministry ran from 1996 all the way up to 2018 with the Angler Diary program. Again, a bit of a finicky data set. It relies on angler recalls, reporting, self-reporting in a book. But we also found that through that survey, angler catches were hovering either around a sort of a mean, which is not indicative of anything. It's indicative of a healthy population. It's not increasing or declining that you might expect if there was some kind of impairment on it. So again, don't expect you to believe me. Without showing you the data. So this is our the top line here is our real information, very sporadic. This is part of the issue with this data set, but it's also honestly the best long-term data set we've got for this within the river. And the bottom here is our annual diary program. So this is an annual program again from 1996 to 2018. So you can see here compared to the 70s and 50s. That even the 90s that are more recent creel surveys were quite high for walleye catches. And then here, if we look at it on an annual basis, we can see that the trends are sort of hovering in and around the mean, which are the dashed lines here. With a spike up in 2018, which correspond to a very similar spike that we were seeing actually like around the same time. Something happened, something really happened. We walk the wall, consistently, including small bass. Again, very similar story. More recent time period, catches above the long-term mean. The lower catches like half. And then sort of our annual summary, our annual survey, either around sort of hovering in around a mean, which again is not indicative of the population increasing or decreasing. It's fairly stable. It's relatively stable. Again, we feel that the subcriterion comes in. On to the final subcriterion number 5, that there is evidence of ongoing and increased spawning activity for fishes since 2006. So I'm going to show you this part of this. I'm going to tell you right now the evaluation against 2006. It was quite different. 2006, I believe that time was picked because that was around the time that some of these construction projects started to occur. But it was also around the time that a lot of surveys started to pick up and look for spawning within the river. So there's a bit of a founder effect here. So we didn't really find evidence pre and post that time period, but we did find significant evidence of spawning activity in the river. In fact, we found so much we had to start breaking it down and looking at it different things. Evidence of adult spawning. So we found evidence of papers adult lake sturgeon, whitefish, walleye, yellow perch, northern madcom, and muskellunge. We know through tagging studies that muskellunge are returning to the river every year in the spring. In fact, Michigan relies on those returns for their muskellunge hatchery program. So we know that they are spawning in the river. When we looked for eggs and larval fish, they found them pretty much everywhere. In fact, they found 41 different larval species with a fairly steady diversity level over time. So way back to around 2007 time period when these studies started coming out, right up to sort of the more recent ones in the 2015 range. The other thing we looked at was we look at trying to find some evidence through genetic papers. So what genetics are going to tell us is sort of what happened in the past before we look at this. And is there any evidence of sort of, again, population size dwindling and becoming so small that there's a lasting genetic effect within those populations. So these studies are not, there's not a ton of them. There's one on walleye, one on yellow perch, a little bit on lake sturgeon here. But in all three of those studies, we found diverse populations with potentially even subpopulation structure within the river. So that means we've got pockets of sort of self-reproducing groups throughout the river, which is indicative of a healthy system. So here again, no evidence of any type of impairment, and we believe that the subcriteria has also been met. So to conclude, as I walk through this, we you too, or thought too. Through all of our research, all of our literature review, all of our review of the agency assessment reports, we found solid evidence to meet each one of these sub-criteria. And we believe that there is no signs of impairment within fish populations within the Detroit River. That said, based on the package, just want to clarify, fish populations is numbers of fish. So we did have some questions through the Public Advisory Council. We are not specifically talking about tumors or deformities. There is a different beneficial use impairment for that. We are talking about numbers of fish and self-reproducing populations in this case. So just wanted to put that one out there because it was a great debate. So that's what I got for you this evening. I will happily take questions.
So yeah, does anybody in the room have any questions or online? If you have one online, just raise your hand and unmute your mic and then yeah. Oh, sweet. If you want, yeah, go ahead, say your question. Just try to speak up so that way the microphones can pick you up so people online can hear.
You said that the spawning of fish actually increased. What would have caused that?
So I did because I don't actually, I have no direction on spawning. Because we don't actually have stuff we don't really have any assessments pre 2006. A little bit before 2010, but part of what went on in the river was the restoration of those reef sites. So again, when that channelization went through it destroyed a lot of that natural spawning habitat, and that was one of the emphases of trying to recreate and restore some other spawning sites. They were specifically designed for lake sturgeon. But through a lot of the assessments, but they found walleye spawning on them. They found lake whitefish eggs on them. They found burbot eggs and on them as well. So burbot's a cold deep water species that they have at east of the lake historically. They found evidence of burbot spawning in the river as well. So the restoration of some of these reefs are likely attributed to if there is actually an increase, but we don't actually have the data, we don't have a time series data set to speak to an increase, we just know there's a lot of spawning. Thanks for the question.
Okay, does anybody have any other questions? No,
Anybody online?
I don't think so. Awesome. So I think that pretty much wraps up the presentations and stuff for tonight. So again, if you guys have any questions, comments, concerns that you didn't get a chance to ask, the QR code is up on the screen. You can put those through online, or obviously you can come up and ask any one of us. But yeah, thank you all for coming. We really appreciate it. And yeah, we look forward to fully redesignating these BUIs in the future.